So today I'm going to talk mostly about what I've seen in the lab this season. And then I'll add a couple extra little tidbits at the end that I threw in just so that we could go over it in case you see it or you have questions about it. So I want to start by talking a little bit about the plant disease triangle. So the reason I'm doing this is because it's very, very important, in particular for disease management, to understand that disease is caused by the coming together of a susceptible plant, a virulent pathogen, and a conducive environment. And so, in this is infectious disease. There are other diseases that are abiotic. We sometimes call them disorders. And so that is due to some environmental factor that is unbalanced or affecting the plant in an unusual way. But again, to go back to infectious disease, I want to bring your attention to the adjectives that I'm using here. So the host needs to be susceptible for disease to happen. The pathogen has to be virulent. So not all pathogens are going to affect all plants. And some can more aggressively affect some plants versus others. And then that conducive environment, which is where we have some control when it comes to management, but a lot of the time we also have no control because the weather is ever changing and we don't know what we're going to get. This year being a really good example of that. Okay, so the way I'm doing this is I'm dividing or I'm presenting the diseases that I want to show you in by pathogen group. And so we're going to start with fungal and oomycete diseases. Oomycetes are pathogens that are like fungi, but they are actually in a completely different kingdom, and they usually like very wet and cooler most of the time environmental conditions. And so here I'm showing the first one, which is, early season, we might have a problem with damping off. So we did see a lot of this season. And so commonly the fungi that are responsible for this are Botrytis, Fusarium, and Rhizoctonia. And then we have two oomycetes that also contribute to damping off, which are Pythium and Phytophthora. So these pathogens are soil-borne, and they can be found in most soils just across the world. But for damping off to develop, the conditions do need to be moist. Usually it's a lower temperature. Sometimes there's an interaction with soil properties. So, for example, if you have soils that have high soluble salts, then that stresses the root systems and that leads to infection by these pathogens. So they like to attack weaker plants, hence them attacking plants that are little. And that might be, just rooting, just germinating from a seed or just being transplanted into the field. pH can also matter for the development of this disease, and depending on the pathogen that's present and causing this problem, that we can use pH to manage the issue. However, to be able to do that, we need to know what it is. But when you see that little plantlets are not growing, they're stunted, or there's wilting, or there's yellowing in the leaves, then my suggestion would be pull that plant out and see if you have a healthy root system or not, because a lot of the time you will find that the root system is just completely gone. Sorry, I had to turn the phone on here. Okay. So next I want to go over black root rot in strawberries. This is caused by a complex, just like damping off, of different pathogens. There are fewer, And most of the time, or at least here in Maryland, I've found Rhizoctonia and Pythium to be involved, but there are other fungi, mostly fungi, that can be associated with black root rot. And then a lot of the time, the black root rot complex also means that we have root lesion nematodes present. So what happens? Well, if there is, again, environmental stress, and that usually means poor drainage or a lot of variability in watering. So poor irrigation management, or there's some harsh drought conditions that are occurring in the spring or in the fall, soil compaction, maybe uneven fields with low areas, then all of those factors can lead again to root stress. If nematodes are present, that adds an opportunity for creating wounds in the roots. And um, so these lesion nematodes, they actually will migrate, or we call them migratory nematodes, which means that they go in and out of the roots. And so they will go inside of the root. They'll enter there with their mouth part. That's called a stylet. It's like a little syringe that they poke in, and then they'll enter the root, and they might suck nutrients from different root cells there, and then they'll come out. And so to reproduce and to, move on to another root. So that wound that they create allows for fungi and oomycetes to enter roots more easily. So a lot of the time, because of the presence of these nematodes, what we see is these patchy, I'm not sure, can you see my pointer? I forget how to make the pointer a different color, but I'm hoping you can see my mouse pointer here. And so we have these sections of roots that are black. So if they coalesce, if it gets bad enough, oh, thank you, Hayden. Then we might see the whole root turning black, but a lot of the time we see these sections. And so what happens is, of course, that the plants don't develop properly. And so we see smaller plants that wilt very quickly, yellowing, sometimes they turn purple. And so they're just in very bad shape. Okay, let's move on to the next one. This is another strawberry problem. We see crown rots in strawberries often. And so there are actually 3 different pathogens that can cause crown rot. One is Colletotricum, which is a fungus, and it causes this anthracnose crown rot that you see here on the left. This tends to be a brown discoloration. When you cut through the crowns, it tends to be a brown discoloration. It has it's described as a cinnamony color, but it really depends on what the moisture level is and the variety of the strawberry and the intensity of the infection, and so severity of the infection. But of course, this leads to poor nutrient uptake. Now, what I'd like you to know is that unlike these other images, you can still see whitish roots here. The root system is a little bit more developed than what we have when we have a Phytophthora infection. And so in the case of Phytophthora crown rot, which is also called red steel, because of a reddish coloration that it can develop, the roots here tend to be rotten as well. And so mostly at the top. And so again, when these crown rots occur, then plants tend to look, the foliage tends to look very stunted and poorly developed. Conditions for Colletotricum are hot and wet. So when it's hot and wet, we might have this problem. It can survive pretty well in debris, and it's spread by water splash to other plants. So even though it is infecting the roots, it enters usually from the above ground portions of the plant. Phytophthora, on the other hand, is a soil-borne pathogen, like I mentioned when we were looking at damping off. And so, and it's more easily infects plants that are under environmental stress. Poor drainage is also a key factor that influences the development of crown rot due to Phytophthora. And so with Phytophthora, another thing to keep in mind is that we might see plant collapse later in the fall or early in the spring. So when the temperatures are warming, not necessarily when they're hot. Okay. Here is the third pathogen that can cause crown rot, which is Neopastilidiopsis. And so if you work with strawberry, you might know that Neopastilidiopsis also causes leaf spots and fruit rot. And so we'll start with the crown rot over here. So it's a brown reddish crown rot. Unfortunately, it looks a lot like the Calotetricum and the Phytophthora crown rots. And this can be a latent infection. It's usually comes into fields from infected transplants, or if there's a history of Neopastilidiopsis in the area, then a leaf infection can lead to debris that might survive where the Neopastilidiopsis fungus might survive. On the leaves, we see leaf spots that can look pretty generic, but they are often surrounded by these reddish discolorations. Although that is also a color that, because it's strawberry, it tends to have this reddish discoloration when disease happens. And then we have this fruit rot here. So actually, I think I'm noticing, so I apologize. So this one here below where it says leaf spots and fruit rot, that here is Neopastilidiopsis. This one in the center is actually Calotetricum. And so I guess even though I did this incorrectly, it's still a good opportunity to note that it's very hard to distinguish these pathogens. Okay. Importantly, for managing Neopastilidiopsis, we really need to use clean planting materials. So even when there has been some pressure of Neopastilidiopsis, if it's not too severe or not too high pressure, really using clean materials can make a big difference in the next season. All right. Another fungal disease that I want to talk about is early blight of tomato. And so I mentioned this because, and I don't know, Hayden mentioned that he was walking around. Well, I had the opportunity to go to a few vegetable farms last week, and I saw so much early blight. I was surprised because, again, I mostly work on ornamental plants, although my heart is in vegetables, but there are very few diseases in ornamental plants. It's mostly abiotic problems that we're seeing, especially in the landscape. But in the case of vegetables, I think the hot and humid weather, which really is a key factor for most of these diseases, has really affected plant growth and health this season. So with the leaf spots, early blight of tomatoes, symptoms of the disease are leaf spots and stem necrosis. So you might notice that the leaf spots have these concentric rings. So we call it like a bullseye target spot. And so the same happens on the stems where you see the necrosis. Now, this doesn't happen 100% of the time, but it's a good characteristic to pay attention to if you're scouting or looking for trying to figure out what the fungal problem is that you have. And then another important characteristic of this characteristic of this disease is that it develops symptoms kind of from the bottom up. And so the reason for this is that the leaves on the bottom can be in contact with contaminated soils, and they also, that moisture that is held in that bottom portion of the plant when there isn't pruning that's done on the bottom, retains enough humidity for the fungal development to occur. Slowly it'll move up, and it can actually cause total plant collapse in severe cases. The fungus, Alternaria, there are different species of Alternaria. It can survive in plant debris. It can also come in transplants. Maybe lesions are very small, but it's very important to look for them before you put your plants in the ground. And on occasion, it can also survive on seeds, not inside the seeds, but on the seeds. Okay. Last fungus that I want to show you is white mold. And so this white mold, or in the case of tomato, it's also called timber rot. And so this is a common problem in fields, in greenhouses, and high tunnels, but more in greenhouses and high tunnels, or where it's very hot and humid. So again, hot and humid is the common denominator for a lot of these problems. It also affects many other vegetables, and these are some. So beans, cabbage, lettuce, pepper, tomato, it doesn't really distinguish too much. And so a couple things to look for is early on, the transplants can show necrosis at the soil line. And then if the conditions are humid or very moist, if the soil is wet, if there's a lot of canopy, humidity, and moisture, then it can develop the soft rot. And so slowly, or it will girdle the stem, and then the rest of the plant dies. With the soft rot, it might be a slow progression to like straw colored stems that kind of collapse. And also, we might see a lot of this white fluffy mycelium. And so not always will we see the mycelium not always will we see the mycelium, but we can see it on the outside and the inside if we cut through that stem when the conditions are right. As the disease progresses, which can happen very quickly, actually, depending on those environmental conditions, the fungus will start to develop these structures called sclerotia. So these are overwintering structures that can then be shed into the soil. So if, for example, this plant isn't removed and quickly, then that as it falls over, those sclerotia can move or can remain in the soil for many, many years. And then you have continuous white mold problems in your fields or in your greenhouses. Okay. So I'm going to show or present some recommendations for management. So I know these are a lot of words, but I put them all up here because I know that this is recorded and then posted. And so I figured this was a good place for you to have some of these tips for managing fungal diseases in vegetable and strawberries. And so scouting is going to be key. So the sooner you can find a problem, the easier it is to manage it. Whether or not that means taking the plant out or, knowing that you have that, it will reduce the inoculum available next season if you catch it early. It's very important to use clean seed in transplants. That means that you get them from a reputable source or that you use resistant plants when possible. And so not all crops have developed resistance to all of these pathogens, but there are resistant plants for some of these diseases. You want to avoid moving infested materials and soil from an infected field to a non-infected field. And so that even means when you're working or whoever's in the field is working, it's better to start with a field that is healthy or less, showing fewer symptoms, and then move into a field that has more symptoms. Um, it's important to remove plants that look very dead. And so if they've wilted and they are rotting, it's better to just take them out and destroy them. For some of these pathogens, it's fine to incorporate into the soil, but or compost, but for some of them it is not, like white mold. Um, so uh, so again, best to just discard them. Um, Managing the soil conditions is going to be key. And so soil conditions and also the conditions around the canopy. So again, hot and humid weather tends to be a common denominator for a lot of these diseases, but more than heat, it is the humidity. And so fungi and oomycetes really like humidity. Oomycetes actually like where they need free water. So when it's very wet, that's when they thrive. So soil drainage is going to be very important to manage. It's important that we reduce leaf wetness periods by irrigating in the morning or irrigating at a time when we know that the plants have enough time in the day to dry out. Spacing I know can be hard to manage because we have to balance that with um, yield efficiencies. And but when possible, that's a good strategy to use to be able to manage these fungal problems. And again, scouting will help because if we do find um, symptoms of fungal diseases early, then that helps us, that allows us to use some fungicides that are available. There are preventive fungicides and there are um, treatment fungicides so that we can use to for these diseases. And the vegetable guide that is produced for the Mid-Atlantic is a very good resource for finding formulations that work for the different pathogens. One other thing to keep in mind is that crop rotation will help a lot. Well, plant selection is important. And so for some of these pathogens, um, we can reduce the amount of inoculum that is present especially for the soil-borne pathogens, but it's important that we don't put in plants that are also hosts for those problems. Okay. I'm going to stop very briefly to see if there are any questions. See everybody at once. No questions. Okay. Also, feel free to add them in the chat if you like. Let me see. There are no questions yet. Okay. Hayden, you can help me with the chat, I hope.
Yeah, I'll keep an eye on the track. I guess one of the questions I had, when irrigating, is it best to, and you have to do it during like a period that it's very wet, is it best to do it in midday, late morning, late kind of day? I know you don't want to do it. You kind of want the water to burn off, but out of the worst possibilities, kind of which is the best one?
I think the morning is better because if you do it midday when it's very hot, then you don't get a lot of the water going into the soil or really, um, yeah, water will be less available for the plants because it's drying so much faster. And so it's better to do it before it gets very hot. So whatever time that is, but before it gets hot.
Okay.
And so morning is better because we are able to get some of that water to penetrate the soil so the roots can take that up and the temperatures will go down and that helps the plants when it gets very hot. So does that help?
Yes, thank you.
Okay. It also will lead to, okay, there's enough moisture that the plants can, take up the water that they need and the nutrients that they need. But then also as the day warms up, that leaf wetness period will be reduced, or, we're minimizing that because it's getting warmer and the leaves are drying. Okay. So let's talk about some nematode diseases that I've
We
seen.
do have a question in the
Oh,
chat.
okay.
If you'd like to answer that from Julia.
Okay, limited spacing and rotation options, but I've had white mold on green beans. Oh, yeah. So unfortunately, Julie, the white molds can, and let me go back for a second to the white molds. Nope. Okay, so I don't know exactly if a mustard cover crop, and I don't know, Hayden, maybe you know better, will help with white mold, but I know that white mold also infects brassicas. And so I think um this is a hard question, especially if you've had this for many years. There is an option for biological control that's expensive. It's called contans. But if you have other growers that you know, it is possible that you could buy it together and kind of share that resource because you're gonna buy a large amount and you won't need to use all of it. There are also, if you see white mold, it is really important that you take those plants out as soon as you can so that these sclerotia are not left in the soil. And so let's say that you already have some collapse, you want to kind of take up some of that soil as well. And then I can add some resources or send some resources to you too um to kind of help with managing where, yeah, it can be difficult because you've had a history. And so depending on the crop, that might look a little bit different. And so we can talk more with an expert and maybe there are some options for you that will work better. I hope that helps a little bit. Mhm. Yeah. Yeah, that's a good idea. Investigate contents. Okay. All right. So I was at nematodes. Okay. So I just want to remind you that I did talk about nematodes at the beginning of when I was talking about black root rot in strawberry. So I'm going to show you that slide very quickly. So here we were discussing root lesion nematodes that again are nematodes that are quite aggressive in causing lesions and root rot. And mostly because once the wounding is there, then there's an opportunity for other pathogens to come in. And so that's one that we see commonly in strawberry, but also in different vegetable soils and even corn and other field crops. And so another one here is root knot nematode. And so I think you all know about root knot nematodes, but one thing that is important to know is that really whenever you see a wilted plant, I know it's hard to say I'm going to pull this out, especially when it has some tomatoes on here and it's producing, but it is a good idea. Let's sacrifice one so that we can figure out what the problem is. Root galls can be very evident or they can be pretty tiny. So there are some examples of both in this image. So here you can see, yes, the root galls are evident. There's, these like round structures that are in enlarged portions of the root. Even here, there's an enlarged portion. But on this root, for example, you see just, the root isn't smooth. It is a little bit lumpy. And so on occasion, it looks like that. Um, I know that doesn't help you very much, but if you suspect that you have a nematode problem, we can help you in the diagnostic lab to see if we can find some nematodes and or do a soil test. Virginia also has a nematology laboratory where they can look for nematodes and do a population count. Above ground, what this looks like, because you're not pulling your plants out all the time. So how would you know that you have root galls? What you find is stunting plants, so plants that are not growing properly, um, that maybe it's a retarded growth compared to other plants around. Usually in fields, these plants occur in pockets. And so we don't have a uniform nematode population across the field, but we have kind of groupings of nematodes in the soils. If you have nematodes and you're tilling, preparing the soil in different ways, or if you have this problem in high tunnels where you're, moving soil around quite a bit, then you might you might be seeing a more uniform effect of the presence of the nematodes. Leaf chlorosis is also evident. So this might look a little bit like a nutrient deficiency because it is. So the roots are not able to take up nutrients. Inside of these galls is a female nematode that has just decided to make its home there forever. And so what the nematode does is it pokes a root cell and it actually sends some signals into the root cells and says, okay, now I'm anthropomorphizing here. So like they don't talk to each other, but um, you plant are going to make nutrients for me instead of making roots for nutrients for yourself. And those signals also create a little bit of what we may consider a tumor. So cell proliferation and enlargement. And those cells are dedicated to producing food for that female. As that female enlarges, it actually will produce eggs inside, and then eventually it will the juveniles will come out and continue to infect the the plants. And so again, it looks like a nutrient deficiency because the roots are not able to take up nutrients. And because of that deficiency, and also lack of uptake of water, then we also see wilt and we can see some foliar death. Okay, those are two main nematode problems that we have. So I want to tell you a little bit about nematode disease management. So much like with the fungi, it is important to use clean seed and transplants from reputable sources. And so it's also very important to sanitize, especially if you know that you have a nematode problem, or if you share equipment with someone that you know has a nematode problem, it's best not to share or to sanitize thoroughly before you use the equipment or the tools. When you start plants, just like if you do it yourself, instead of obtaining it, grow seedlings in a nematode-free soil. Crop rotation is going to be very good for nematode management, but it is important to select plants wisely. And so here's a short list of plants that can um, suppress those populations of nematodes in the soil. In tomato, for example, there are plants that are resistant to root knot nematodes and there are resistant stocks as well. Weeds are important to manage wherever you're doing this, in the field, in the greenhouse, in the high tunnels, because weeds can also be hosts for the nematodes. And so you might, rotate, but if you keep weeds there all the time on the edges or wherever, then, you have a problem because those populations continue to be there. There are nematicides that are available. They are not going to kill everything, but they can help if you do all of these other, or, use some of these other strategies for managing the nematodes. Okay, I'm going to do bacterial diseases before questions, and then I'll stop for questions again. So I've also seen bacterial diseases in my walk and in the laboratory this season. Two very common ones are bacterial speck and bacterial spot. So bacterial speck is produced by generally Pseudomonas syringae. And you'll say, well, what's the difference? They all look like spots to me. They all look like spots to me too. But the bacterial speck, I guess a speck is a smaller spot. And so it just looks like little tiny flecks of necrosis on the leaves. And then bacterial spots can, well, before I move on, as the disease continues to progress and the bacteria continue to infect and spread even on the same leaf, then we get coalescing lesions that will cause larger areas to die. And so that's where it can get confusing with bacterial spots. That where the bacteria do arrive on the leaf and infect, but cause a larger lesion. For both of these, actually, you can see fruit symptoms, but in the case of xanthomonas, it's usually more prevalent. And so you see these little necrotic areas on the fruit. Sometimes they will be accompanied by halos and sometimes not. Yellow halos is what I mean. So there is a lot of yellowing that is around a lot of these specks and spots, which can help in determining or diagnosing this as a bacterial problem. But depending on the conditions, those yellow halos may or may not form. This season, they are present. And so you want to check for this. Now, at this point in time, there's probably nothing to be done. It's unfortunately, we plant pathologists have not quite figured out how to control bacterial diseases effectively, but there are some products that can help, and I'll show you that in the next or in the management slide. Bacterial or sorry, leaf wetness is going to be key in managing leaf wetness is going to be key for these diseases because bacteria do like, again, hot and humid. And so the longer there are bacteria, or sorry, the longer there's water on the leaves, like what we were discussing with the irrigation, then the more the diseases will spread. So this is where, again, scouting really matters because if you catch this early, then you're able to apply something that could help suppress the population and not have so much necrosis that might lead to some fruit quality problems. The other disease that I've seen this year is bacterial canker. And so it tends to cause fewer leaf lesions, but it can, however, a characteristic symptom of this disease are these necrotic spots or lesions on the stems and the petioles. And so slowly they again coalesce and cause a larger lesion on the plant. And if you can observe here, these edges of the leaves are a little bit necrotic, and that might look as though, and also the necrosis might look as though it is a nutrient issue on occasion, but you see that the rest of the leaf is quite green and so again, hot and humid, believe it or not. This tends to be the pathogen comes in seeds. It's seed transmitted very efficiently in pepper and tomato prevalently. It can also come in transplants. And so it's important that you use clean stock too. In the tomatoes, on the fruit, the lesions will look scabby and have this whitish appearance. And the bigger problem, I think, with this versus the Pseudomonas and the Xanthomonas is that Clavibacter can survive for a long time on debris. So the others will, as the plants collapse and they are decomposing, the bacteria will die because they require this living plant, but Clavibacter is more resilient to those changes and can survive for longer periods of time. And so it is important if you see this that you catch that early. And if you know that you have this in your field, that you use some preventive measures in the next season. Oh, it's raining. Okay. All right, bacterial disease management. Again, scouting is going to be important. Using clean seed is going to be very important too, and transplants. And all of these can be seed transmitted. So I don't think that it's common practice to save seeds for tomato production. But if you produce heirlooms and want to save your seeds, then if you see these problems, I highly recommend that you start with a new source of seeds. You want to, again, avoid moving from infected fields or highly infected fields to cleaner fields. And this is because the bacteria that are on the leaves, if the symptoms are severe, well, even if they're not, they can come on you and you can spread them. Rain splash and rains with wind can very easily spread the bacteria. And so it's important to try to minimize that splashing when possible. So a leaf wetness period I already mentioned, and then sanitation is going to be important because again, the bacteria can be transmitted very easily. And in the case of Clavibacter, if there is any mechanical um, processing of the plants after harvest, then it's going to be very important to sanitize equipment. There are some resistant plants that can be used for bacterial diseases in general, but sometimes that they're hard to find. Okay, I stop here for any more questions. Oh, yeah, marigolds. I have a question from Jan about marigolds. Marigolds can they discourage nematodes? That's a good question. So what, um, so the plants that are used for rotating for nematode management, can have mostly two functions. One is that they're non-hosts, so the nematodes won't reproduce in them, and so they'll die over time. And second, that they might secrete some compounds uh, that can affect the survival of suppress the nematode populations. And in the case of nematodes, I am not sure. But if you have a nematode problem, planting marigolds near tomatoes is not necessarily going to help you with the nematode issue. I think you need to substitute the tomatoes with something for some time. It can help because if you don't have a tomato right next to a tomato, then the population growth is less. Okay. All right. It is almost one, and I knew that that was going to be the case because I'm a little bit of a talker. And so I just wanted to very briefly, I quickly threw in a couple of slides because I wanted to share these with you. That they haven't been except for the virus diseases here. The others I have not seen them, but I did see them last year. And so I think it's important to share. So virus diseases are very common and they're mostly associated with insect vectors. And so you want to make sure that if you see any symptoms, and this could be necrotic spots on the leaves, it could be ring spots on fruit, it could be malformed plants, which can also be confused with herbicide damage, or mosaic and crinkling, then you want to look for a pest. So not all of these viruses are transmitted by insect or arthropod pests, but a lot of them are. So in the case of cucumber mosaic virus and um, uh, some of the other um, cucurbit viruses, they are aphid transmitted. And so if you have aphids, especially if you're in an area where there are a lot of wild plants or forests or something, then you could have the transmission of viruses into your field. Or if you know that you have a field, let's say alfalfa or um, a grain field, like a wheat field or something near you, and they've harvested, then if there are aphids or other insects there, they will like come out of there and move into your crops. And so it's important to keep out and make sure that you're scouting for pests and that you're using appropriate pest management strategies. Um, some of these other are thrips transmitted, and few of them are mite transmitted. Some can be beetle transmitted too. Okay, close the chat here. The other group of diseases that I wanted to show you were fruit rots. And so botrytis fruit rot is quite common in strawberries. And this is also going to be mostly managed with moisture and also some fungicide treatments, although there's a lot of resistance in botrytis. And so I know that there was an earlier webinar. I'll guess, I think it was Neopestalotiopsis, but my colleague here, Mingjun, who studies botrytis, and so he provides regular updates on what is the best practices for management. Pythium and fruit rot can also occur, especially during these times. And again, if the environment is wet. And then we also can get the occasional bacterial soft rot. So this looks white, but I think there are a couple of things going on here. So I think there's a fungal infection, but there's also a wet rot. And a lot of the time, bacterial soft rots, Pectobacterium or Indicia for other crops, what happens is that they enter through very tiny wounds on the surface of the fruit. It can also be in other plant parts. So like in potatoes, this is also common. In sweet potatoes, it happens. In dahlias, it also happens. And then they start rotting from the inside out because they prefer anaerobic environments. And so, or shouldn't say anaerobic, but very low oxygen environments. And so they will rot, and it might not even be smelly. Sometimes it is. Okay, and the other is anthracnose. The weather helped us a little bit early in the season, but these might pop up for harvested fruits. And so anthracnose tends to be, yeah, kind of sunken lesions that may or may not have this like targeted area. And often you will see fruiting bodies. You don't really see them here, but fruiting bodies that will show up if the environment is moist enough. And then finally, there's some abiotic issues that we all keep an eye out for. And this is more of, early season type things to look out for. But if we do get any events, hail events later on, then hail damage is usually, you can observe it a few days after the event, and it can lead to fruit rot. So it's important to protect your crops if you know there's going to be a hail event, if you can. And then the other thing is herbicide injury, which early in the season is a huge problem. There can be herbicide residues in soil. There can be drift from other areas. There sometimes, if you're in a greenhouse or in a hoop house and that's where your production is, if outside of the hoop house there are herbicides that are applied, then it can still come into your house and affect your plants. The other is mulches sometimes. If you use mulches to help with weed suppression or keeping moisture in the soil, they can on occasion also have herbicide residue. And so you might see these symptoms of curling, and they look very viral, but if it's not too bad, the plants will grow out of it, kind of like what you see here. So this plant has grown out of these malformed stems. Okay, last thing here, very little time, but is how can you submit samples to the plant diagnostic lab? So I have a couple of steps. So step one, you can visit our website or you can contact me. You can scan this QR code if you have the ability to do that in the next few seconds before I change my slide. But you can also just Google Plant Diagnostic Laboratory at the University of Maryland and find us there. Step two is to read the sampling submission instructions. So I say this because it will help to decide which samples to send. Ideally for vegetables, if you can send the whole plants, that is great. Sometimes tomatoes are really large. So if you're not sure where the problem is, if you send like the top and some fruit and then the bottom, then that is ideal. And in a little bit, I'll show you some pictures about how to package that up. Step 3 is to complete the submission form. This is really important because, as I mentioned, there are a lot of environmental conditions and practices that can lead to disease. And this is something that I don't know. So when I get a sample, I don't know where it's growing or how it's growing or, what is the variety and what you've applied. And so it is very helpful to me to know how you're managing your plants so that I can provide a better diagnosis. Images are super helpful. So along with your diagnosis, you can email pictures. And these are some really good pictures that were taken by a crop scout. And so, we're looking at adventitious roots over here with some death of the stems. Then the stem was cut. And so it's revealing discoloration, both severe and mild. In the case of this person, they pulled out a few plants. And then, what does the root system look like? It looks pretty decent. It doesn't, things like that. And so whatever you can send picture-wise would be great. I also appreciate it when I get pictures that are showing the full planting because it helps with looking for patterns that might indicate what the problems are. This is how we like things to be packaged. So you can send them in a Ziploc bag. And then if you're sending soil, separate that. You don't have to put the submission form in a bag. But if you're concerned with things being wet, then you can do that. And then for roots and soil, it is very, very good to wrap them in either a bag or aluminum foil because the aluminum foil stays closed because the soil will, in transit, everything moves. And so it'll get all over. And then it makes it a little bit hard if we have to culture something. It actually also causes plants, especially if they come from hot environments, to rot a lot faster. So last one, I'm leaving this up, and that's just our contact information. And I did not leave a lot of time for questions, but I'm happy to answer them if you have them.